Okay, I have my timer started here. I see everyone is settling in with their free lunches, and I just want to say this is my 10th year at PEGS. Very excited to give this talk. It's great to see some familiar faces and meet some new people, like-minded individuals. Before I start, just wanted to talk about the elephant in the room here. There's a lot of controversy around AI, but I think one thing we can agree on is that there is no such thing as a free lunch. So I can see here today, you all have your free lunches, but you're paying with your attention. It's a beautiful day outside, and you could be out there. So I appreciate your time, and I'll try to not take it for granted. I think today my goal is to kind of show my excitement for AI in drug discovery, and hopefully we'll all learn a little something today. So my talk is really going to focus on how we utilize artificial intelligence for immune repertoire mining and multi-objective engineering. But for those of you who are not aware of Ailux, a little bit of a brief introduction. Ailux is the biologics division within XtalPi. XtalPi has been around and a global leader in AI drug discovery for over a decade, and we're a smaller team within the larger XtalPi. So our team is around 85 people, and you can see from this graphic here that 2/3 of our team are actually wet lab experimental scientists, and 1/3 as an AI company are computational scientists. And this team is all under the same roof, and they work hand in hand with the wet lab data feeding into the dry lab to make our proprietary algorithms. We've performed over 80 different projects across discovery and engineering with about a 50-50 split there. We've had quite a bit of track record of success, working with over 30 different partners, and recently had some releases showing that we've licensed some of our algorithms to J&J and UCB, which we're very proud of. And one thing personally, as an alliance manager working directly on projects, I'm proud to say that 75% of the partners that work with us after initiating end up expanding into the larger scope for those projects. But today I'm going to introduce our three main workhorse AI platforms. We have a whole host of different AI algorithms, but these here are our three main platforms. CrystalFold is our structural modeling complex prediction algorithm. ZenProt is our generative AI platform. That's a large language model. And Zentient is the predictive AI suite, which can predict wet lab data directly from sequences alone. But all of these proprietary algorithms are really fueled by our Atlas database. This is an extensive database that has larger antibody sequences than available in the public domain, and we've spent a lot of time and resources internally building out to a higher quality of data, including developability and affinity values for all of those sequences. And we are modality agnostic. We work with IgGs, VHHs, bispecifics, ADCs, TCRs, and most recently and excitingly, peptides. All of this being supported by our comprehensive wet lab. So let's jump right into CrystalFold. I'm sure everyone in this room is familiar with AlphaFold. CrystalFold is a structure prediction algorithm. So you put in the antigen sequence and the antibody sequence, and in about a day, our algorithm gives a high resolution predicted complex structure that you can use for a variety of downstream applications. As an antibody engineer, it gets really exciting because you get that sequence information and that structure information at the earliest stage. So you can do a whole host of different things with it, from epitope identification, early lead optimization. You can predict function, and then also identify pairs that play well together for bispecific design. A little bit more detail here for CrystalFold. So it is industry-leading protein complex prediction algorithm. Here we have some data that compares it head-to-head with AlphaFold Multimer 2.3. On the left-hand side, you can see that when we evaluated 39 different complex structures, AlphaFold Multimer 2.3 had a 44% success rate, whereas our algorithm, CrystalFold, had a 70% success rate. And AlphaFold 3, although it is not publicly available, In their white paper, they described a 63% success rate when evaluating 65 different antibodies. So what do we mean by success? Within this field, one of the characteristics we're looking for is a DocQ score above 2.3. So the DocQ score is a simple algorithm that compares the output predicted complex structure to the ground truth structure. So that PDB structure that you see on the right-hand side is the bona fide crystal structure. In these three case studies here, crystal folds significantly outperformed the other guys. And you can see here that the very high DocQ scores suggest that we were able to identify and generate a high resolution structure compared to the PDB structure. Okay, so moving on to our other AI platform, ZenProt. ZenProt is, I like to think of it as the ChatGPT for antibodies. So it's a large language model that has learned the alphabet of proteins, and we utilize it in a whole host of different ways. Very similar to ChatGPT, some of the ways that I use ChatGPT, I'll put in a bad sentence that I've written, and then it'll give me a good sentence. And that's kind of what you can do with ZenProt here, taking some hits and leads and asking it to improve developability of these molecules. And we utilize it here, and I have two different case studies here from examples where on the left-hand side, you can see a molecule had issues with immunogenicity. This molecule, natalizumab, is famously known for failing in the clinic because of the immunogenicity. A paper came out in 2019 which identified some point mutations that improved immunogenicity, but it actually reminds me of my time maybe 10 years ago doing antibody engineering. You solve one problem and you can create a whole host of other issues. And you can see that here with the mutations that were suggested in the 2019 paper. They were able to improve immunogenicity, but they took a massive hit on yield and other developability parameters. With our large language model, we can now ask the algorithm to make suggested mutations that improve all areas simultaneously. So this is truly multi-parametric optimization. And we did this again and again, and here's another example here where a molecule had issues with polyreactivity, and we were able to improve polyreactivity and improve all of the other developability parameters simultaneously. And finally here, I just want to introduce Zentient. Zentient is very exciting for me as a former lab scientist. This is the predictive AI suite. So with sequence information alone, it's predicting wet lab data for a whole host of developability parameters and affinity. So as you can see here, we have a high accuracy for predicting the wet lab value for affinity. So we can just from sequence information alone predict the affinity of that molecule. And then we can also predict over 15 different developability characteristics, again, from sequence information alone. So we truly are having in silico screening at this stage. And we also utilize Zentient for repertoire discovery. We can use Zentient to pull out hits, potential hits from an NGS data set, and it can scrape at a higher rate and identify 5 times more hits than traditional human-driven approaches. But really, all of this wouldn't be possible without our Atlas database. This is a proprietary database that our team has spent quite a bit of time and resource curating, with the real intent of balancing both the quantity and the quality of the data set. So you can see from some of these metrics, our database is 5 times larger than publicly available databases with the heavy and light chain pairing. We also have significantly more unpaired antibody sequences. And then we've utilized our wet lab to pair actual developability and affinity data with those sequences to have a comprehensive, high-quality data set. And we also are continually probing our own algorithms to determine if we need more data. And you can see here from these plots that we started with a data set that had 500 molecules, and then we retrospectively pulled some of that data out and did the same algorithm and learned on 300, and then also on 145. And you see that with more data, there is an increase in performance for these algorithms. But it's not just the quantity of the data, it's also important to look at the quality of the data. And maybe quality isn't the right word because I'm talking about negative results. It's often challenging to find in the public domain negative results for a sequence. I can hear the ghost of my Ph.D. professor telling me that negative results don't get published, and that seems to be the case, especially for clinical molecules. So we've intentionally spent resources in our wet lab to make sequences that we know have problems so that we can generate and balance out positive data and negative data. So that's the way that the algorithms learn best, from seeing both the positive and the negative. And all of this really is the foundation on our wet lab. As I said, 2/3 of our team are wet lab scientists, and our computational team really relies on them for generating the data to make our proprietary platforms. We have all the toys that you would need: binding kinetics, flow cytometry, any manner of cell-based assay. We do our protein production in-house and then do all the quality control, biophysical, analytical analysis. And all of that is leveraging automation so we can increase throughput. And we also have our own cryo-EM to support the structure modeling work. Okay, so let's jump into some real data that I have. I have a few case studies that I'd like to go through that really demonstrate how we create unique value with our AI platform. So as I said, we're modality agnostic, and we've done a whole host of different case studies that I'd happy to share with you. We can connect later, but today we just have enough time to go through three main areas. One around antigen design, hit identification, and then finally multi-parametric optimization. So for my first case study, very hot topic, right? GPCRs. This was a case where we were identifying an antibody for a difficult to express GPCR. As you all know, these molecules are not happy. They lack stability and they're not really well expressed. So that was the main issue with this wild type GPCR. So our plan here, as you can see, is to take the wild type GPCR, and at first we take that sequence and go into our ZenProt large language model. The generative AI actually in silico designs over 160,000 different variants. So 160,000 different variants of this wild type GPCR. So we really are exploring a very large sequence space. It ranks those molecules based on developability, and we take the top 10,000 of those molecules, and it goes into our more refined Zentient predictive AI platform. So with those 10,000 sequences, Zentient can predict a whole host of different developability characteristics, predict affinity and developability. Again, ranking those molecules, we take just the top 100 of those molecules into a much lower throughput model, standard molecular dynamics modeling, to really find molecules that have the best stability. And then at this point, we finally get to expression. So I just want to let that sink in for a second. All of that work before this moment is all in silico characterization and screening. Finally, we get down to a set where we can express in the wet lab. And we only have to make 15 molecules because of all that pre-screening. From this specific case, we made 15 molecules, and 11 of those were validated. And what do I mean by validated? They were well expressed, thermally stable, and obviously correctly folded. And I think one of the most interesting things for me from this case study is not only that it only takes 8 weeks, but that there's over 100 different point mutations in the transmembrane domain and the intracellular loops in the AI-designed mutants. In order to present the business end of the GPCR, you really truly are establishing a fully new scaffold. So here's a little bit of the wet lab data from this specific case study. As you can see from this SDS page result, the wild type had very negligible amount of expression, but here 2 of the mutants had significantly improved expression. These AI-designed variants were very thermally stable. They were challenged at 40 degrees for 10 minutes and maintained their stability. And most importantly, they were correctly folded. So here there's an ELISA. This is a competition ELISA. You can see a dose response curve as we're able to compete off a benchmark antibody from the wild type GPCR with our mutant variants, confirming that we have proper folding. So after all of this quality control, what do? We go into an immunization, into an animal, and here we have the data for titer results from that immunization. So this is special here because we're generating the immune response with the AI-designed mutant variants, but then titering on the wild-type GPCR which is expressed on cells. Now, this was a small case study, so we wanted to close the loop. So for this small-scale screening, we only pulled out 13 hits, but all these hits had pretty good cell binding, as you can see here. And then most importantly, one of those 13 was cyno cross-reactive, and it had comparable function to benchmark. Now, one cyno molecule out of this bunch might not sound very impressive, but when you compare it to the clinical benchmark antibody, the clinical benchmark antibody in their publications showed that it was not cyno cross-reactive, and it had over 300,000 hybridoma clones just to get that one hit. And just saying 300,000 hybridoma clones makes my thumb hurt from all the pipetting that was probably necessary to screen these numbers. So when you're thinking about the decreasing amount of resources that we use by screening with AI, I think we also need to think about the thumbs that we're saving from grad students and scientists. Okay, I'd like to move on to our second case study here. This was an antibody discovery campaign to generate an antibody to a very specific region of the TCR. I would just like to start out by warning everybody that I am not an immunologist, so please be kind. But the idea here is that we wanted to raise an antibody against a very specific region of the TCR. So as you can see, the TCR here has both an alpha chain and a beta chain. We want to raise an antibody to that boxed region that you can see on this slide. That boxed region includes CDR1 of the beta chain and CDR2 of the beta chain, which are largely invariant. The majority of the peptide contacts come from CDR3 and its hypervariability. But those invariant chains of CDR1 and CDR2 on the beta chain is really what we were targeting. I'd like to talk a little bit about how we do this process. We utilize another one of our AI platforms called Spotlight. This Spotlight is really focused on antigen design. So we wanted to focus the presentation of this invariant region of the antigen while obviously maintaining stability and performing in a way that we have the proper conformation of that beta chain. So it starts out with the parental TCR of the desired B beta family. We do 20 different AI designed variants, and the AI platform is suggesting mutations within the region of the non-human regions to maintain proper stability and conformation of the region of the human region that you can see in this graphic. So the whole grayed out region is non-human region that will hopefully be silent to the mouse during immunization. After we've done the quality control, that antigen goes into the animals, raises an immune response, and then we take that into our Trilux hit generation platform. So our platform is quite extensive. We have a 3-in-1 platform. You know, each of these different platforms for identifying hits, they all have their strengths and weaknesses, and we've learned that using them, 3 of these in parallel really creates a diverse set of antibodies. So we have our single B-cell screening platform, which we call Smart Fluidics, and then ExploreSeq is our AI-based Repertoire NGS analysis tool. And then SpeedPlay is our immune phage library, which allows for a very interesting, unique biopanning. From this specific case study, you can see the number of clones that we pulled out. So here we had over 140 different sequences. And what's unique that we recently started doing over just the past year is a process of high throughput humanization. So we take the mouse sequence at that earliest stage as soon as it's identified as potential binder, and we humanize those molecules in one day. So we use our AI platform in conjunction with some other expert knowledge, and we're able to humanize these molecules at the earliest stage once we have those sequences. And the significance of this is that we don't have to express the chimeric antibodies anymore. So we don't express the chimeric antibodies and then slog through a screening triage to get to your final hits, and then go back, humanize those final hits, and then have to repeat all of that work to confirm that activity. We just humanize right from the beginning. From this specific case study, we had 143 humanized hits, 28 of those were agonists, and 12 were V-beta specific. But for this case study, just to prove that we were doing the right thing, we also did a parallel effort, a full-blown parallel immunization campaign using the wild-type TCR, just to see how much better that our AI designs operated. And we did get one binder from the wild type TCR activity. But you can see here 12 times more specific binders justifies all of that screening work and in silico work up front. I'd like to take a little bit of time, kind of go through each of these sections individually. So our Spotlight platform, which is really antigen design and focusing the epitope of that antigen so we can raise antibodies just against that region. It really starts with the parental TCR. We do some structural prediction and evaluation and identify several different non-human scaffolds. Once we have those non-human scaffolds, that gets put into our AI algorithm, and we get multi-parametric stability prediction. So we end up with around 20 different AI design variants, and that's the stage where we go into the wet lab. We make those 20 different variants. They go through small-scale expression. We take the top AI design variants and move those forward for immunizations. Once the immunizations are complete, we go into our 3-in-1 Trilux platform. Again, these 3 platforms all have their own strengths and weaknesses. Smart fluidics, single B-cell screening is a droplet sorting technique, and then we follow that with single B-cell NGS. ExploreSeq is our AI platform that can scrape NGS data. So ExploreSeq is a pre-trained multimodal predictive AI that performs at a significantly higher rate than traditional techniques for evaluating NGS data. And then finally, SpeedPlay is our immune-based phage display library, which allows for very unique biopanning. Again, these 3 working in conjunction allow us to identify very diverse clonotypes that you can see represented here from this plot. There's very little overlap in sequence between these 3 different platforms, and we're regularly pressure testing this to ensure that it's worthwhile to consume these resources across 3 different platforms. And then I just want to touch on the high throughput humanization one more time. So this is really an affordable and really time-efficient approach and a great alternative to transgenic mice. You're getting humanized sequences at the very earliest stage. So we get our murine mAb sequences. It goes through a process of framework selection, and then we use our generative AI platform to guide the back mutations necessary to humanize these molecules. And then at that stage, they go into expression. So right now, we can do about 500 antibodies and humanize those in one day. And for expression, we really only have to express 1 or maybe 2 different variants per parental mAb to get a 90% success rate. So 90% of the molecules that go through are fully humanized after the fact and maintain affinity. And I think one of the most interesting things about this platform is that it works across species. So you can do immunization for mouse, rat, rabbit, camelid, and then as soon as you get those hits, they all go into humanization together and then can all be characterized together. So it really increases that head-to-head nature of putting antibodies from different species up against each other and letting the best come through. Okay, moving on to my 3rd and final case study here. This is around IL-6 and generating a selective GP130 variant. Again, not an immunologist, but we'll give it a try. So GP130 FC, these molecules exist. The goal here for GP130 FC, as you can see, there's molecules in phase 2 that are acting as a capture for IL-6 because IL-6 is implicated in chronic inflammation. But IL-6 and IL-11, both binding partners of GP130, they have normal processes within the body, and we don't want to impact regeneration or host defense. So the goal for this project was to generate a selective GP130 that would only selectively bind IL-6 and not bind IL-11. And as I said, there's a molecule in phase 2 right now that is a GP130 FC, but it's not selective, so it may have some impact on regeneration. And of course, there's the IL-6 and IL-6 receptor antibodies, again, not selective. And this is kind of a tall task, I think. So if you look at the wild type binding affinities for GP130, both to IL-6 and IL-11, they're very high affinity interactions. Our goal, our goal for this work is to get a threefold increase in GP130 binding to IL-6 and a tenfold decrease in binding to IL-11. And of course, we want to maintain developability and have patentability with unique novel mutations. So this whole process starts with crystal fold. And I can say that about a majority of the work that we do. The foundation of our work is really around crystal fold and predicting the complex structure. Now, on the left-hand side, you can see GP130 and IL-6. That complex structure was already publicly available when this project began. But the complex structure for IL-11 and GP130 was not available, and we had to utilize crystal fold to generate that structure. We use these structures to identify the key contact residues between the GP130 and the cytokine. And for this specific example, there were 110 contact residues between GP130 and IL-11, which really suggests a very large space for engineering. So how do we go about this to get affinity modulation? We use a process of active learning in conjunction with our AI models in a design, make, test cycle. So we start out with that predicted structure, and then we go into ZenProt, the generative large language model, which will make suggestions on mutations to get to our desired location. All those suggestions go into our pre-screened predictive AI model, Zentient, and then the top of those molecules go into our wet lab for expression and testing. And this is a cyclical nature because all of that wet lab data then goes back into the algorithm, both the positive and the negative data, so that ZenProt can learn from that and make improved suggestions. So here's the take-home message from this effort. After 4 rounds of affinity modulation, we actually yielded 7 different IL-6 selective variants that were all developable. These were GP130 variants. And the way this data is plotted, these little dots moving up and to the right are good, and you can see in the top right-hand corner here, there were several variants that fulfilled our TPP. And these are all color-coded so that you can see from round to round, we continue to get progress moving towards our target product profile. If we go through each one of these rounds very quickly, it starts with crystal fold, generating the structure model, identifying those key contacts. As I said, this is a massive space from an engineering perspective, 110 to the 20. And the generative AI ends up identifying 2,000 different single point mutants that could potentially get us where we need to go. Of those 2,000, they're pre-screened in our Zentient platform, and that suggests the top 45 prioritized mutants to make in the wet lab. We express those molecules, test those molecules, and that data goes back into our algorithms as an active learning step for subsequent rounds. And you can see from round 1, we don't really see a lot of movement. I think, this reminds me of my time in the wet lab. You're making single point mutations, you oftentimes don't really get a very significant change in your antibody. At times you can, but this data really is helping to support the subsequent rounds. So this is kind of what round 2 looks like. In round 1, we've identified and we have that wet lab data. It goes into our algorithm. After that process of generative AI, now it's making combinatorial mutants. In this time, round 2, we have up to 3 mutations per GP130. It ended up being 5,000 combinatorial mutants. And that pre-screening in Zentient prioritizes the top 96 mutants. Those become expressed. We test those, and then we continue with that cyclical process of active learning. And here you can see round 2. We're starting to get movement towards our target product profile, but it really is not enough just yet. And I'm just, for the sake of time here, showing you round 3 and 4. Going forward, even further increasing the number of mutations for our combinatorial mutants, in this case up to 9 combinatorial mutants. The mutants are pre-screened, prioritized. You go through expression and characterization. The scoring process and machine learning allows us to continue to improve. And in this specific case, we had 7 different hits that fulfilled our TPP. And you can see them represented here. And it's interesting to me that even round 3 pulled out a few hits that were sufficient for the TPP. And all of these molecules were developable and compared to benchmark. So in summary, we have been able to use our 3 different AI engines and an active learning step in this design, make, test cycle, all supported by crystal fold protein complex structure modeling. For this case study, we had 4 rounds of optimization, and we only expressed 233 variants and identified 7 different patentable variants. But we don't do this just for proteins. This is an example for antibodies. In this specific case, for an antibody, we did 2 rounds of optimization. We only expressed 65 different variants, and we had a 13-fold increase in affinity, and it took just 5 weeks. So just in closure here, some of the lessons that we've learned over the years. We've had the luxury of having the wet lab and the dry lab all under the same roof, and that really has lots of synergies. We intentionally are balancing the quantity and the quality of our training data sets to maximize what we can get out of our AI platforms. And we're kind of tool agnostic. We utilize any tool that we can to get to where we need to be, and because in the end, it doesn't really matter about the AI tools. It really is about making the best drugs. So we're always interested in meeting new people who have new ideas and partnering with any exciting technology. So please come and find us. We have a booth, and I'll be in the back for any further questions. So thank you.